Hello there, Erickson and Dylan here. We're excited and ready to talk about mitosis and meiosis. Starting off, we would like to talk about mitosis, the division of genetic material and the contents of the nucleus into two complete separate cells. So, before cell division, mitosis goes through a really big phase called the interphase. During interphase, the cell carries out its regular metabolic functions, and it goes through three phases, G1, S, and G2. During G1, rapid growth and metabolic activity happen. During S phase, DNA synthesis and replication. And during G2, cell prepares for division. After G2, the cell is ready for mitosis. The first phase of mitosis is prophase. During prophase, centrosome duplicates to form two centrioses, and after separation, each pair migrates to opposite poles of the cell. Chromatin threads, shortens, and becomes visible as two sister chromatids join by a centromere. During metaphase, chromosomes migrate to the center of the cell, or the equatorial plate, and spindle microtubules attach to the centromeres, and the centromeres divide. During anaphase, the centromeres divide, the chromatids move to opposite poles of the cell, and the sister chromatids split. During telophase, two cells have been formed. The nuclear membrane reappears, the spindle microtubules disappear, and this causes it to restart the cycle. Now I pass it on to Erickson.
Thanks, Dylan. Meiosis is the process that produces haploid gametes from diploid cells in the ovaries and testes. What makes meiosis different from mitosis is that it goes through two nuclear divisions, meiosis I and meiosis II. But before it goes through a nuclear division, it goes through interphase. After interphase, it starts on a process known as prophase I, and in that phase, the nuclear membrane begins to dissolve. The spindle fibers form and the chromosomes come together in homologous pairs. Also, they'll come together in synapse. Oftentimes, the chromosome mingles up, and when they mingle up, they found different homologous pairs and break, exchanging segments in a process called crossing over. After the homologous pairs cross over, the spindle fibers attach to a centromere of each chromosome as the chromosomes line up along the equatorial plate in metaphase I. As they line up, the cell goes through anaphase I. The homologous chromosomes move towards opposite poles. This is called segregation. Reduction division occurs, and one member of each homologous pair will be found in each of the newly formed cells. This leads up to telophase I. A membrane begins to form around each nucleus, and the two chromosomes in the nucleus are not identical. Each of the daughter cells contains one number of the homologous chromosome pairs. The chromosomes are similar but not identical. As telophase I ends, the cell division doesn't stop. Instead, it goes through meiosis II, in which in prophase II, the nuclear membrane dissolves and the spindle fibers form again. In metaphase II, the arrangement of each chromosome within each chromatid aligns along the equatorial plate, and the chromatids are held together by the centromere. In anaphase II, the chromatids of each chromosome break apart and migrate toward opposite poles. Now the chromatids are called chromosomes. Lastly comes telophase II, where the second nuclear division is completed. It causes cytokinesis, and then the 4 haploid daughter cells are produced. With the 4 haploid cells, if you are male, you produce 4 sperm cells. If you are female, you produce only 1 egg.
Thank you for watching and hope you have gained more knowledge about mitosis and meiosis.